I guess everyone is here. It's past 12. So as usual, the seminar series is co-sponsored by the P41 Funded Center for Mesoscale Mapping, housed in the Martinos Center. And today we have the privilege of hosting Dr. Michael Wheeler. Dr. Michael Wheeler is an assistant professor of neurology at Harvard Medical School and Brigham Hospital in the Jean Lay Institute at Anne Romney Center for Neurologic Diseases. His research background is in neuroimmunology and neurobiology, focused on the role of neuroimmune interactions that control neurodegeneration and behavior, as well as developing new tools to study these interactions. The Wheeler Lab applies a systems neuroimmunology framework to behavior, identifying the molecules, cells, and circuits that underlie changes in brain-body communication in neuropsychiatric disorders, addictions, and inflammatory diseases. So please join me in welcoming Dr. Michael Wheeler.
All right, thank you so much. Okay. So then so all set, hide this, right? And then good. Okay, great. Yeah, thanks again so much for the invitation to be here today. And so today I'm going to give you a little bit of introduction to our work studying the potentially neuroimmune properties of psychedelics and how these can recalibrate maladaptive behavior and stress disorders and other diseases. And I'll give you a little bit about some of where we're going with this work in terms of trying to understand the precise role of immune cells in psychedelic-induced plasticity, which is kind of ongoing in the lab. So just to start with a broad introduction. So in my lab, we're a collection of immunologists, neuroscientists, studying the brain, basically. And so in our lab, we try to think of the brain actually as another type of immune tissue, like a mucosal tissue, kind of the same way an immunologist would think of the gut or the lung. And the reason for that is because as we learn more and more about the neuroanatomy of the brain, we understand that it's actually constantly exposed to signals from the immune system. So the brain is surrounded by fluid and there's fluid pulsing through it, CSF and also blood. There are immune tissues that immediately adjoin the brain, which include the meninges. And there's actually conduits from meninges, skull, etc., for immune cells to get into the brain. A lot of these immune cells that live in these tissues are long-lived. There's virtually every class of immune cell in these tissues. And this is just in homeostasis, by the way. And the brain can actually drain these signals and convey them to peripheral organs through the lymphatic system. And so in this way, we sort of view the brain as constantly exposed to these immune signals. And our hypothesis basically is that these immune signals are profoundly shaping the function of the brain. And so the kind of way we view the brain is that it's a mucosal tissue, a type of immune tissue, if you wish. But instead of respiration or digestion or something, its function is that it controls behavior. And so in the context of disease, this becomes really important because if these barriers get disrupted, if the abundance of these immune cells changes, then you can start to influence behavior and so on and so forth. And we know that in the context of multiple diseases and also during homeostasis, the brain is constantly interpreting different signals from the immune system. So in the context of Parkinson's disease, we know that there's types of T cells that can react to brain antigens, potentially causing degeneration. There are senses from the periphery, interoception, that can actually convey signals of what's going on in peripheral tissues like the liver, the gut, the lung, back to the brain, where those signals are then converted into neural impulses. And we know that in the context of something less severe than neurodegeneration, like stress or depression, there actually becomes a leakier blood-brain barrier where actually peripheral signals and sometimes cells can get into the brain, and then again, they start to modify this crosstalk. So really the key hypothesis is that neuroimmune interactions in the brain and other tissues are important in the context of physiology and also disease. And so one of the areas that we've been really interested in is the context of mental health and in particular depression. And I just include this slide to emphasize how common it actually is. So it's roughly 50% of people experience this in their lifetimes. It's across countries, it's probably underreported, and it's across different sexes. And so, and it's across age groups. And so it's kind of a huge problem. And then one of the challenges that we face in this area is there's been a lot of amazing work done studying the neural circuits that convey these signals, from one neuron to the other. But we really don't have great therapies that target, let's say, other types of interactions in these diseases. So, for example, if you look at this summary of all the therapies that are available to target neurotransmission in depression, they all target basically the synapse, or many target these kind of receptor systems. But there is evidence that in depression and also other types of psychiatric related diseases, there's changes in the peripheral immune compartment and also the central immune compartment. So this is just a meta-analysis demonstrating that in the periphery of MDD patients, patients with depression, there's this elevation of IL-6 production. And in some patients and some studies across decades, treatment with SSRIs, if the patients respond, actually lowers some of these peripheral cytokines. And there's also evidence that these cytokines are playing a causal role, at least in some preclinical studies, in modifying circuit function. And so in this way, we sort of take the approach of thinking our mental health disorders really just disorders of brain-body communication is that one aspect that gets disrupted. And so broadly, what I'll talk to you about today is how we're trying to study stress-induced neuroimmune interactions as a potential way to better understand the circuits that are dysregulated in mental health disorders. And then I'll kind of tell you a little bit about some ongoing work about how we're thinking about these molecular and cellular networks in the context of other models. And so one of the questions we had is that, we know there's this association between peripheral immunity and changes in behavior, but is there a way that we could systematically identify mechanisms, cells, and time points that are co-regulated as a way to potentially link changes in the immune system with changes in behavior? And so Lizzie Chung and Jinsu Lee in my lab. So Lizzie was a post-bac student. Now she's at UCSF, and Jinsu's a postdoc in my lab. They undertook this series of studies where we were trying to understand over a longitudinal time frame, what is the stressor duration that evokes changes in, let's say, stress responses, the HPA axis, but then also immune system changes and also behavior and also changes in immune cells themselves. And so is there a time point where we can detect all three coincidentally regulated? And so we subjected mice to a 3-week paradigm, or 1 to 3 week paradigm, where we performed chronic restraint stress and then subjected them to this fear acquisition and fear extinction, fear generalization type of assay. So kind of your classical conditioning, they get exposed to a shock with an unconditioned stimulus, or excuse me, a conditioned stimulus. And then we just expose them to, let's say, the tone of the light, and we see sort of how fearful they are. And when we did this, we found that at 3 weeks, but really not earlier, we detected this increased exacerbation of these freezing responses when we switched them to a new context. We detected the same type of behavior when we subjected them to the elevated plus maze, which is kind of a different measure of this fear behavior. So I'm sure you all know how it works, but half of this maze shaped like a plus sign is closed, the other half is open. If the mice spend more time in the closed arms, then they're considered to be sort of more fearful, more anxious. But on top of this, we also detected elevated production of cytokines in the plasma of these mice, suggesting that at the time point where we detected changes in fear behavior, we also detected changes in cytokine signaling, immune system signaling. So we wanted to dig into this a little bit more. And one of the cell types that we've been very interested in the brain as an interpreter of these immune signals are astrocytes, which are these brain resident glial cells, as I'm sure you all know. And they're unique in the sense that anatomically they have these very long wispy fibers. There's hundreds of them, it seems to be the current estimate, where actually some of these processes actually directly wrap the vasculature, while other processes, as you can sort of see here, are embedded into synapses, and they're so closely embedded that they actively recycle neurotransmitter and so forth. And so in this way, we sort of view astrocytes as these fulcrums of neuroimmune interaction. So they can sense what's going on in the periphery almost, at a very rapid rate. And they can transduce these signals within the cell and also potentially convey them to the synapse, and also vice versa, right? They can potentially sense what's going on in the synapse and transmit it to the vasculature. So we're very interested in these cells as transducers of neuroimmune signals. And so after we determined that around 3 weeks was this time point where there might be this coincident regulation, we generated an astrocyte-specific reporter mouse strain where it expresses a fluorescent reporter under an inducible astrocyte-specific promoter. We subjected them to this 3 weeks of chronic restraint stress, and then we performed single-cell RNA-seq of the labeled cells. And what we found is that at around 18 days of stress, there was this subset of astrocytes, transcriptionally defined from this analysis, that was relatively more abundant at the stress time point relative to controls. When we went in and dug into it more deeply, we see that there's, simply more of the stress cells in this cluster, and we really thought it was potentially this stress-associated population. Molecularly, we wanted to dig into this a little bit more to provide clues about, what these astrocytes might be expressing and how they might be involved in the stress response. And so when we looked into it, we found that the glucocorticoid receptor, which is the key mediator of stress responses, was highly activated in these cells and predicted to regulate this population. And beyond glucocorticoid receptor signaling, we also saw, which was upregulated, we saw downregulated signaling by a family of receptor tyrosine kinases that are all linked by this molecule called EGFR, which is a receptor for EGF and other molecules, and I'll tell you about it in a second. And on top of that, these astrocytes projected along a trajectory were associated with sort of a late stage activation state, which we thought might be representative of their involvement in stress responses. And beyond the pathway level of analysis of EGFR activity, we also detected decreased activation of EGFR in this stress-associated subset, and also downregulation of EGFR in other subsets of astrocytes, suggesting that it might be more broadly relevant than just this transcriptional population. And so what is EGFR? It's a receptor tyrosine kinase. It senses a variety of different ligands that have different affinities and so forth. They're produced in a state-dependent way by multiple different cell types. EGFR is thought to be broadly anti-inflammatory. So, for example, the production of amphoregulin, AREG, in the gut is known to dampen inflammation to some extent, and also in the context of stroke. And we were intrigued by EGFR as a candidate regulator of these cells, because in a previous study where we developed basically a genetic screening platform using droplet microfluidics, we found that astrocyte interactions with brain-resident macrophages, microglia, were strongly controlled by EGFR signaling. So if EGFR signaling was perturbed in this network, which is kind of the microcosm of neuroimmune interactions, the cells got more inflammatory. And in fact, that's just showing you activation of an NF-kappa B pathway in cells that either were targeted with a non-targeting guide or where EGFR was deleted. And these signals were driven by TNF and IL-1 beta, which are the same cytokines we detected as elevated in the plasma of the stressed mice. So we thought there might be a common mechanism by which EGFR and astrocytes may limit local inflammatory responses and potentially limit stress behavior. Because actually several studies have shown now that if you start to boost inflammation in neural circuits, it acts as kind of a driver of those circuits. And so this would kind of explain the fear behavior we observed. So one of the questions we had was, our analysis of astrocytes was brain-wide. So where would EGFR expression be most impactful? And the way we approach this is EGFR acts as a negative regulator. So lower EGFR expression and therefore increased inflammatory responses might predispose certain brain areas toward activation. And consistent with that idea in our fear behavior data, we analyzed both a single cell RNA atlas and stained on the protein level all across the brain. And we find that the amygdala, the fear center of the brain, actually expresses the lowest quantity of EGFR in astrocytes. So it suggests that if there's a kind of this increase in inflammatory tone, like what we detected in stress, but potentially the amygdala is less well equipped to deal with it than other brain areas because there's less EGFR astrocyte expression. That was our hypothesis. And I'm sure everyone here knows all about the amygdala, probably much better than me, but the point is that it's actually a series of interconnected subnuclei. Some are inhibitory neurons, some are excitatory neurons. They all interproject. But broadly speaking, the lateral and basolateral amygdala are thought to be the input nuclei. They're excitatory. Basically, if you activate them with optogenetics or something, then you basically drive fear. If you lesion them, then you have no more fear. And so we thought that perhaps astrocytes in the basolateral amygdala could be driving these excitatory neurons and promote fear behavior. Um, and so what we did is we performed a knockdown approach of EGFR expression specifically in astrocytes. And then the idea here is that we subjected mice to restraint stress in the same sort of behavioral assays, but rather than at 18 days, we did it at 1 week after restraint stress, which is actually a time point where we didn't detect these potent behavioral changes, and we also didn't detect strong immune system engagement. And we thought that this would be akin to sort of putting the circuit into overdrive. So deleting a negative regulator at an early time point might actually just recapitulate what we saw at 18 days. That was sort of our hypothesis. So we had Cas9 under the control of the astrocyte-specific promoter together with a guide RNA against EGFR or a control guide, and then we bilaterally targeted the amygdala. And what we found is that if you knock down EGFR in amygdala astrocytes, you actually do recapitulate the 18-day stress phenotype, which is that the mice are more fearful. So elimination of EGFR in astrocytes at an earlier time point is actually detrimental. And we validated this also by elevated plasmase, and we did a similar analysis using EGFR flox mice and bilaterally delivering a Cree virus. So basically, you can knock it out anyway, and loss of EGFR is actually detrimental. And so what we wanted to understand next is how EGFR expressing astrocytes might be influencing the local microcircuit in the amygdala. Because, to us, we don't necessarily think that the astrocytes are the output transducers of behavior. We obviously think that what they're doing is communicating some sort of signal to neurons. And so we wanted to better understand what might be going on in the astrocytes in this sense. And so we performed RNA-seq of the astrocytes where EGFR was knocked down, or the controls. We detected induction of first of all, of molecules known to stabilize fear behavior in astrocytes that have been previously reported, as well as additional activators of inflammatory signaling, including NF-kappa B and IL-1 and so forth. But one of the more interesting hits that we didn't know much about at the time were these receptor type protein tyrosine phosphatases, which are actually transmembrane receptors that engage with another type of receptor on a neighboring cell. And so of all of the receptor protein tyrosine kinases, or phosphatases, excuse me, in these astrocytes, we detected PTPRS as the most upregulated in our stressed astrocyte cluster that we previously examined by single-cell RNA-seq. We went ahead and independently validated this and found that this PTPRS molecule, which again appears to be showing up in the context of chronic stress, is upregulated in the BLA of mice that were stressed for 3 weeks. And we wanted to validate whether the cytokines we detected in the plasma were actually involved in modulating this process, and we found that the answer is yes. So if you treat primary astrocytes with the 3 major cytokines we detected as elevated in the plasma of the stressed mice, we see an induction of PTPRS expression. And likewise, if you treat these astrocytes with corticosterone and IL-1 beta in the presence or absence of EGFR, if you knock down EGFR, then you also boost PTPRS even further. And so the idea here is that EGFR acts as an anti-inflammatory brake on astrocytes, and it limits the expression of this molecule PTPRS, which has previously been implicated in plasticity and things like that in neurons. And so we hypothesized that PTPRS, more expression of it on the astrocyte surface, might be actually stabilizing some of these fear behavior phenotypes that we detected. So we wanted to understand how that actually happens. And so what we did is, from the same animals where we knocked down EGFR in astrocytes, we also profiled the neurons in the amygdala. And we examined what changes happen in these neurons when astrocytes, when EGFR is knocked down in astrocytes relative to control. And we also see kind of the same engagement of this pathway by receptor protein tyrosine phosphatases. But one of the receptors we see elevated on these neurons is called SLIT-TRAC2, which is a partner of PTPRS, and it's expressed on these neurons. And indeed, if you look at the RNA-seq data and the expression, SLIT-TRAC2 is one of the top candidates that's expressed on these neurons when EGFR is knocked down on the astrocytes. And again, the way this sort of works, I think I have it. Yeah, the way this works is, at least in neurons, for example, is presynaptic and postsynaptic neurons will express one or the other of these proteins, and they can engage, which sort of stabilizes neurotransmission. And so in the astrocytes, although we don't really know, which part of the astrocyte it's expressed on, and same with the neurons, we hypothesize that the engagement of these two receptors that can interact might just stabilize astrocyte neuron interactions and facilitate the activity of these excitatory neurons that drive fear behavior. And so that was our hypothesis. And so on top of that, we wanted to understand if this pathway is engaged, what might it be engaging in the neurons? And so pulling from the same neuron RNA-seq data set, we identified transcription factors that are induced in these neurons in the absence of astrocyte EGFR. And this is kind of the list of the top 5 that comes up. And this just shows it a different way. And so what we did next is we co-cultured astrocytes together with neurons, and we knocked down either of these components alone or together. And what we find is that among the top candidates that we then sort of filter this list down, some of the top candidates that are engaged by PTPRS slit-track2 astrocyte neuron signaling are NR2F2 and this molecule CEBPG. And some of the other candidates we didn't really focus on. And just to cut a long story short, we started to focus on this molecule NR2F2 because it is reported to play a role in the amygdala in the context of stress. And we also did a knockdown screening platform to knock down each of these factors in neurons. And I'll show you some data on NR2F2, but CEBPG didn't seem to be playing a large role. And so, as I mentioned, because our data suggests that this astrocyte neuron interaction might be controlling this NR2F2 transcriptional program in neurons, we developed the same sort of AAV system to knock down NR2F2 or control in neurons bilaterally in the amygdala. And so the question we had is, if this is a way that excitatory neurons are becoming more activated, let's say firing more, something like that, might that just be also involved in just producing more machinery for synaptic contacts and things like that following fear conditioning? And that's more or less what we found. So if we express M-cherry in excitatory neurons and knock down NR2F2 or control, we basically see that the arborization of the excitatory neurons in the BLA goes down, meaning that NR2F2 seems to be kind of promoting this synaptic machinery, if you will, or at least the possibility to engage in more synapses following fear conditioning than the control animals. And again, this is showing the co-localization of pre and postsynaptic neurons following stress and fear conditioning. And what we see is that if you knock down NR2F2 in these neurons, that the fear conditioning behavior, or at least the synaptic readout of the behavior, is dampened. So you have sort of NR2F2 promoting seemingly fewer synapses induced by fear conditioning in the amygdala. And that's also what we see if we look at the behavioral level. So if you knock down NR2F2 specifically in these neurons and you perform the contextual fear conditioning assay, if you delete it, you have lower fear behavior, which is consistent with all of this gain of function of excitatory neuron drive. And you also see sort of decreased activation of pathways involved in stress shown in these two panels. And so one of our next questions is, again, so we're very interested in how all of this is shaped by the immune system or brain-body interactions. And so one of our next questions is, how can we begin to define the regulation of this circuit? So we sort of detect the inflammatory cytokines in the periphery, we detect these changes in plasticity in the brain, but what's really driving this on the molecular and cellular level? And as I mentioned before, the amygdala is not just one monolithic structure. It's composed of many subnuclei with many complex connections and so on. And so we thought that the best way to begin to identify this question was by performing spatial transcriptomics, where we can sort of retain the entire complex structure of the amygdala, but then look on the single cell level at the transcriptional programs modified by stress and fear conditioning and potentially the immune system. So we turned to this approach that's called StereoSeq. And StereoSeq is a very high resolution spatial transcriptomic platform where at the time it was comparable to many of the leading approaches in terms of resolution. So you basically have 500 nanometer resolution, so subcellular, and it's also sequencing based. So we thought that was helpful because then we can examine in an unbiased manner the transcriptional programs induced in different cell types in the amygdala following stress. And so we performed the assay where we stressed mice for 3 weeks or control, and then we subjected mice to fear conditioning and then and fear expression, let's say. And then we profiled the whole coronal section of this brain containing the amygdala by stereoseq. And we subsetted out all the sort of major anatomical landmarks and called cell types using a previous cell atlas that was developed brain-wide and then specifically applied to the amygdala. And this is just showing you all the cell type markers that we detected. So we can detect basically 6 major cell types, excitatory, inhibitory neurons, different types of glia. And these are all the different sections. And we actually get pretty high quality spatial transcriptomic information. And just to tell you, we're also subsetting out directly the nuclei of these cells. So we're actually able to pull out the cell bodies and we don't have to draw boundaries on this tissue. And so we can still get appreciable transcriptional information just by looking at the nuclei. And so this is a composite of all of the single cells that we obtained from the 6 different sections, showing you all the different cell types. And then if we decompose it into one amygdala that we can sort of subset out regionally, we can see sort of the regional distribution of all these different cell types and their relative abundance. And so we did this for all the different amygdala that we captured. And so one of the questions we had is, can we re-identify this NR2F2 expressing population of excitatory neurons in this data set? And so we subclustered both types of excitatory neurons that we detected, and we found that there's, again, a subset associated with this late stage of pseudotime. So transcriptionally, there's a trajectory that these cells are at the end of. And this cluster, which is shown in orange here, that's also the late stage pseudotime, is cluster 2 of the excitatory neurons, and statistically it's expanded, it's overrepresented in the stress conditions, signifying that it might be related. And it's also localized to the excitatory nuclei of the amygdala, including the BLA, which is the input nucleus, and also this area called the N-lot, which is actually a population of excitatory neurons that gets input from the sense of smell. And when we look at NR2F2 expression, which again is this kind of plasticity promoting transcription factor, we think, there's higher levels of expression of NR2F2 in cluster 2 relative to the other groups of neurons. And then we also wanted to determine if this population of cells was interacting with, or if it had anything to do with EGFR expression in the astrocytes. And so what we did is we mapped out the locations of all of the cluster 2 excitatory neurons in every amygdala that we detected in stress. And then we charted what the astrocytes closest to these cells express relative to astrocytes further away. And kind of consistent with the idea that a dampening of EGFR expression in astrocytes may boost the expression of NR2F2. If you look at I think it's hard to see here, so I'll just move this for a sec. If you look at the astrocytes closest to the excitatory 2 subset, we basically don't detect any astrocytes expressing EGFR, but the astrocytes that are further away from these cells express significantly higher levels of EGFR. So it suggests that this interaction may be at play, in vivo in the context of chronic stress.
So was there a correlation between the glial cell types and the location of the excitatory?
Yeah, so I probably didn't explain this well, just to make sure. Tell me if I don't get this right. So this analysis takes the position of every excitatory neuron, and then it calculates the distance to every single astrocyte. And then we said which astrocyte is closest to each of these neurons. And then we have kind of a distribution of the closest neighbor of each excitatory neuron. And then if we subset the astrocytes by the furthest away versus the closest, that's what this analysis on the right is. So in other words, the closer an astrocyte is to one of these neurons in this subset, the less likely they are to express EGFR. That's what that suggests. But if you're asking about things like microglia or oligos, we didn't do that analysis. But we're very interested in these questions moving forward. Did that answer it?
Yes, kind of, because in your previous slide, you had, there was like a map of different,
I knew that was going to happen. Yeah, sorry, go ahead. This?
Yeah,
Yeah.
there you go. And I was wondering, there's a correlation there, especially speaking between where the inhibitory neurons are, or the excitatory neurons, and the different glial cells. That.
Yeah, okay. So we didn't look more broadly than what I just told you about in terms of like overall excitatory neurons. We looked at the subset that we were interested in because we thought it was associated with stress. But more broadly speaking, we are interested in this idea because, you know, we I'll show you this later, but we were very interested to see if any of the microglia we identified were actually recruited immune cells that then sort of matured. So in other words, are some neurons closer to these immune cells that get in and then just stay embedded in the circuit? We're kind of interested in that type of question, but we didn't look more broadly than that on the population level of cell types. Yep. All right. My computer has Diet Coke on it, but it's okay. So that's fine. Okay. Okay, great. Okay, that should be fine. So then, so we're also interested, like I mentioned, in thanks. You know, I really knew it would happen. I'm always flailing about. It's fine, it's fine, it's fine. It's an MGB computer, so it should be. Okay, so we wanted to understand what are the peripheral signals that might be controlling this whole loop. So looking in these excitatory neurons that might be linked with stress, we see many of the same signals we detected in the periphery, including IL-12, IL-1. We see IL-6, which is implicated in other stress models, as well as engagement of NF-kappa B, which is your canonical pro-inflammatory activator. And so we were, as we were just talking about in the Q&A, we were interested to understand, first of all, are these signals coming from within the amygdala, right? So could this just be a local microglia producing these cytokines type of thing? So we plotted the microglia subsets that we detected, and we're trying to determine if these were in fact something like infiltrating monocytes or macrophages or something. And so when we look at their gene expression profiles, they appear to express almost everything you'd expect from microglia, and not really the expression of CCR2, which is a monocyte marker gene. We looked for this gene in particular because many groups have shown that monocytes in particular are recruited to the vasculature during stress. Then we also tried to figure out, are the signals coming locally from the amygdala? And so we looked, again, and this is spatial transcriptomic data, we looked at the total counts of all of the cytokines that were related to what we found in the periphery by our earlier ELISA studies. And we find very, very low amounts of these cytokines produced. To give you a perspective, this is across all of the sections that we analyze, so multiple animals. And, you know, if you look at something like a marker gene of, you know, an astrocyte or a neuron, it's in the thousands. So very, very low amounts. And to just come back to this panel, right, when we look at by ELISA and we see these cytokines, we thought that perhaps some of these signals might actually be, you know, some of the signals might be driving this program because they're proteins coming from the periphery. So our next question really is, what is the peripheral response controlling these properties? And as I mentioned, there's been a lot of work. Really, Scott Russo's lab has kind of led the way in this area, but there's been a lot of work in identifying how peripheral cues, and also Phil Swirsky, I should say, how peripheral cues modify neural circuits in the context of stress. One of the models is that, you know, these cells are recruited to brain areas like the vasculature that's in the brain or something like that. And also they can degrade the blood-brain barrier. So it becomes leakier, but not so leaky as to, you know, require or allow for, you know, full-blown infiltration of many immune cells like you would see in something like multiple sclerosis or EAE. It's just leakier. It's more susceptible to peripheral products. And so what we were interested in is, okay, in our model, when we have these mice that are stressed for multiple weeks, are there changes in the immune compartment following that stress specifically? And one of the places we looked is called the meninges, which I'm sure you all know it, but it's this tissue layer beneath the skull, series of tissue layers. And the meninges houses virtually every type of immune cell. It's involved in multiple classes of immune responses related to the brain. There are ways for signals to not only get from the periphery to the meninges, but also get from meningeal layers into the brain, shown by really pioneered by Yoni Kidnes's lab. And although it's been shown that monocytes can get to the vasculature and adjoining areas, there hasn't been an exhaustive investigation of what's happening in the meninges. So what we see is that actually in stress, pro-inflammatory monocytes, which is this short-lived class of wound-healing immune cells that is, as you would predict, inflammatory, gets into the meninges, and it doesn't really happen. I mean, there's normal baseline levels in unstressed mice, but it doesn't really happen in a subthreshold stress paradigm. And there's actually a decrease in these immune cells from certain peripheral tissues, and this is just shown on the right. And so we wanted to understand, is this monocyte phenotype involved with the behavioral and circuit changes that we see? So what we did is we transferred daily monocytes to mice that underwent this subthreshold stress paradigm, where there's not this inflammation and there's not this recruitment of immune cells to the meninges. There's not even that much of a behavioral phenotype. And so what we can see is if we transfer these monocytes to mice daily, they get to the meninges and we see them in appreciable numbers akin to what we would see at 3 weeks of stress. And when there's more monocytes transferred to the mice show this kind of more fearful behavior in the plasmae shown here. So they spend less time in the open arms. And the same is true in the fear generalization assay, which is the mice that were transferred monocytes show more freezing behaviors relative to the control groups. And what's going on in the amygdala, if we look at astrocytes, is that there are these pro-inflammatory pathways that are engaged when monocytes are transferred. So IL-1, which we know modulates EGFR and PTPRS and all these things, and also decreased EGFR signaling. So that's what happens when you transfer monocytes. We also did the converse.
Because if you look 2 slides back,
Yes.
yeah, it looks like there's like a bimodal distribution.
Yeah.
And so did you transfer the monocytes from the ones that had the high number of cells or just all of those?
Yeah, so that's a good point. So actually, the way we did this is we just took the monocytes from wild type mice. So they weren't primed by stress or anything like that. That's a great experiment that we should do. But we are interested in what you're talking about because we noticed the same thing. And it appears that, there can be some sort of, depending on the animal, we really want to correlate, which subsets of cells are going where and how that links with the behavioral phenotype. But to answer your direct question, we didn't transfer monocytes from the stress mice. We harvested them daily from naive wild type animals and then transferred them to these animals the day before they underwent stress and then daily thereafter. So that's a great point. And actually, if I'm not mistaken, we do have some data showing that if you transfer monocytes to mice that are unstressed, there's maybe like a modest change in behavior, but you really need the stress as well to sort of prime the system, probably to get them recruited to the meninges. So just a second. And so we're also interested in doing the converse experiment. Can we deplete monocytes, particularly from the meninges, and reverse the behavioral changes? And so we injected, we use an injection that kind of delivers products that mimic kind of a meningeal route of delivery. We delivered an anti-CCR2 monoclonal antibody to deplete these monocytes. We can see them reduced even several days later. And what we see is that if you deplete the meningeal monocytes, you can actually reverse the elevated plasmae and the fear behavior changes, suggesting that monocyte recruitment to the meninges is causing, or actually at least more monocytes are causing elevated stress behaviors, and the depletion of meningeal monocytes rescues these. And so, as I alluded to earlier, one of the mechanisms that's sort of been unearthed in the stress field is that the blood-brain barrier becomes leakier. Again, not so leaky as to warrant immune cell infiltration and something very dramatic, but if you can see here, if you inject a labeled cytokine into mice where there's, more or less modulation of the BBB by stress or other mechanisms, there's actually more infiltration. There's more detection of this cytokine in the brain parenchyma when the mice are stressed relative to control. And so we tested this same idea. So first of all, the meninges, I want to just point out, they're underneath the skull, but they really surround the entire brain. And just to show this, we delivered a blue dye into the meningeal space, and just a few hours later, you can see it spread across the entire surface of the brain, dorsal ventral. So it really does kind of permeate everywhere, potentially even into deep brain structures. And this starts to change dramatically when you stress the animals. So if you stress the animals for 3 weeks and then inject a biotinolated version of IL-1 beta, where you can actually now detect it in situ, we detect, some IL-1 beta getting into the amygdala, which is, again, very deep in the brain. But in the stress mice, we see significantly more IL-1 beta puncta in the amygdala than the control, suggesting the BBB is leakier. On top of this,
How big is that on?
IL-1 beta?
Yeah.
That is a good question. I don't want to misstate it, but it's in the maybe I don't want to misstate it. It's in the maybe 10 kilodalton range, something like that. I do forget. But just to tell you, we aren't the first to show this. If you're interested in how wide the BBB actually opens up, I don't have that answer. But we are doing an experiment now where we're injecting different size dextrans into the animals to see what types of molecules can really get in. And Scott Russo's lab has shown very nicely that it really is, and this is his data here, it really is the cytokine or the peripheral products that get in, and not so much the cells. If they look for the cells that are fluorescently labeled, they seem to remain in the vasculature, and that's basically what we see also.
Like 18 kilograms.
18. Okay, I was going to say 10. So I was off by 80%. That's it. Okay, so then so we looked then, astrocytes are also, again, because we detected increased IL-1 beta in the plasma. We looked at, whether astrocytes can sense more of this molecules. Again, since we think this might be rewiring the whole cascade in the amygdala. And we see that astrocytes express more IL-1 receptor in the amygdala. And we also did a follow-up experiment where if we stress the animals and then deliver IL-1 beta cytokine into the amygdala using an injection, we can actually see that IL-1 beta per se boosts fear behavior. And actually, this is something very similar to what Junha and Gloria Choi recently reported for things like IL-17, which promotes anxiety in the amygdala. And so the question then is, if these monocytes are pathogenic, what is regulating them? And so we profiled them by RNA-seq, and one of the pathways that surprised us as coming up is serotonin receptor signaling. And what we did is we went into publicly available atlases and we looked for different types of serotonin receptors. And we found that, indeed, these receptors are expressed by immune cells. And this is actually just a subset of immune cells. In a tissue-specific way, these type 2 serotonin receptors are very broadly expressed across all kinds of different innate and adaptive immune cells. And this intrigued us because these receptors not only bind serotonin, but they can also bind to psychedelic drugs like psilocybin, which is gaining traction for use in clinical trials for depression. And so what we're interested in is, could psilocybin or other psychedelic drugs re-engage pathways in these cells or this tissue microenvironment to modify behavior? And so what we did is we stressed the animals for a few weeks, and then we dosed them with an acute dose of psilocybin or MDMA, which was, at the time, it was also gaining traction in the clinic, but things have changed. But at the same time, MDMA, just to tell you, MDMA and psilocybin, while both kind of similarly applied, are mechanistically different and chemically different, but we wanted to see if there are common mechanisms. And so they reverse fear expression akin to what others have shown. But what was surprising is that when we analyzed the recruitment of monocytes and also other immune cells to the meninges following the dosage with these drugs, there's a complete reversal of the accumulation of these cells to the meninges. So basically what we did is, following stress, we dosed one single dose and looked 1 day later, and you can sort of see just the elimination of these cells recruited to the meninges. And so we wanted to understand, is this a direct or an indirect effect? Because, we are aware, of course, that many different types of neurons, including in the sympathetic nervous system, express these receptors, as well as different stromal cells, things like epithelial cells and choroid plexus cells and so on. And so first we tested whether isolated immune cells in isolation can actually sense signaling from psychedelic drugs. And the punchline is that it seems like they can. So what we found is if we co-treated with corticosterone and psilocybin, there was this kind of engagement of signaling that suppressed certain chemokine receptors. That's largely what we found. But you could reverse this if you blocked type 2 serotonin receptors. Technically, this is a 2A antagonist, but it's possibly dirtier than that. But beyond acting on the immune cells directly, we also took the approach that this might be acting on the tissue itself. And that's because psychedelics are known to drive some sort of vasoconstrictive action in the brain. They're known to alter neurovascular coupling. And so what we did is we imaged the vascularity of the mouse head, basically using a dye that sort of sticks around in the vasculature. And what we find is that following stress, there's this increased vascularization or vascular dysfunction, kind of akin to what others have shown. But if you treat acutely with psychedelics, that vascular signal goes away, meaning that potentially the vasoconstrictive actions of psychedelics might be playing a role. And so we started to try to tease this out a bit more directly. And so we treated animals with psychedelics with or without a systemic vasodilator called nifedipine. And we found that despite the presence of psychedelics, if you then engage vasodilation pathways, you can then boost even further the abundance of immune cells, monocytes in particular, in the meninges following the dosage with this vasodilator. And so what we think might be happening is that psychedelics might be altering a tissue microenvironment where, because of this vasoconstrictive properties, they can modify the space that's simply available for immune cells to stick around and hang out in the vasculature or the meninges. And by at the same time downregulating certain chemokine receptors that are involved in their trafficking and retention, it might promote the egress of those cells out of the meninges or the vasculature into sort of draining tissues. And the last thing I'll say is we also tested the role of the sympathetic nervous system using an ablation strategy. And what we do see is that the sympathetic nervous system does play some important role, because if you dose animals with psilocybin in the presence of a sympathectomy, there's no longer any sort of immune cell egress from the meninges. But as I mentioned before, MDMA and psilocybin are not totally similar compounds from the chemistry side. But so MDMA still has some action independent of the central nervous system, suggesting there's some sort of serotonin signaling that's at play. I can just breeze through this because I don't want to go over time, but we also obtained human cells of human astrocytes. We detected similar engagement of these pathways controlled by IL-1 beta. We also analyzed human amygdala by single nuke RNA-seq. The punchline is we see a similar cluster that has downregulated EGFR signaling. And if we go into the amygdala by staining, we see downregulated EGFR expression in astrocytes from the MDD patients. And something similar holds if we analyze neurons, which is there's a stress-linked population that appears to be at least somewhat transcriptionally controlled by NR2F2. And so to just summarize this, the way we think this is all operating is that stress starts to drive these brain-body interactions, which lead to immune cell recruitment to the meninges, potentially mediated by chemokine receptors like CXCR4, which also happens to be sensitive to psychedelics. Those immune cells, we did have this data, but I didn't show it. These monocytes and other types of immune cells can produce IL-1 beta that may be acting on astrocytes locally in the amygdala to turn on this kind of inflammatory cascade that hijacks plasticity and drives fear behavior. We're very interested in the potential role of microglia because we found in a past study that they produce amphoregulin, which can tune down inflammatory signaling by signaling to astrocytes. And what we find is that psychedelics reduce the accumulation of monocytes to the meninges, and we're interested whether that alters some of these anti-inflammatory pathways as well. I'll just show you one last piece of data so that we can just talk about questions. I'll skip over everything else. But what we started to get interested in is whether the control of the immune system in response to psychedelics is actually playing any role whatsoever in plasticity. So this is very preliminary. But what we did is we turned to another stress paradigm, which is inescapable foot shock, also called learned helplessness. And what we did is we treated the animals with this inescapable foot shock. And then we measured their latency to escape the noxious stimulus. So the way this works is actually the mice that are chronically shocked, they don't even escape when given the opportunity. It's called this helplessness behavior. And then what we wanted to test is whether psilocybin would reverse this behavior. And the trick is that we did this in the presence or absence of monocytes. So we pre-treated the animals with diphtheria toxin or a vehicle, then we did this stress paradigm, and then we did pre and post psilocybin to sort of see what are the behavioral changes in response to psychedelic with or without monocytes. And so this is sort of the treatment regime. And what we find is, again, in this independent paradigm, if you deplete monocytes, then the mice are much better at escaping the chamber after they're helpless. So the mice lacking monocytes appear to be escaping just fine. But what was interesting to us is, and again, we're kind of digging into this a little bit more. What was interesting to us is that if you look at the change in the escapes after the animals are given psilocybin, the mice that have monocytes, the psilocybin appears to be more effective. But when the monocytes are depleted, the psilocybin effect sort of goes away, suggesting that, either the depletion of monocytes sort of boosts the plasticity to a ceiling effect where you really can't kind of engage the psychedelic plasticity pathways anymore, or alternatively, that by reducing immune cell abundance, and again, it's probably not just limited to monocytes, but by reducing immune cell abundance, you're actually also potentially driving this plasticity. So that's something we're interested in exploring further and seeing how generalizable it is. And also, we're very interested to understand the applicability of psychedelic plasticity in other diseases like inflammatory diseases. Just for the sake of time, I'll just skip over this, but the last point is that we identified this molecule, which is called PAX6, in a reanalysis of depressed patient samples from both the blood and the brain. And it appears to be engaged in both the periphery and in the brain, so in peripheral monocytes and also in the brain. And so and the significance of that is we think that sort of common mechanisms are at play that influence the brain and the body and induce kind of similar cascades. And so we're curious if this PAX6 gene, which is sort of co-regulated with CXCR4 and is controlling monocyte recruitment, might be an interesting therapeutic target. And so this is just my last summary slide. So for us, what we think is an interesting model is that, psychedelics are obviously controlling synaptic plasticity. I think that's been shown very clearly by Alex Kwan. But one of the other interesting phenotypes we think is that psychedelics have non-neural roles as well. And because they influence basically tissue plasticity, that might also be impacting the function of the brain. Again, if you're thinking about it as this mucosal tissue that's receiving brain-body signals. And so through these sort of concerted actions, which require many cell types, they may be important for controlling that in other circuits. And so with that, I'll just thank everyone that was involved and played a role in this. And thank you again for the invitation. I'm happy to take questions. Thanks.